Hey, Katie. Hey, Katie. All right. Video off. Hey, Katie, how are you? We're on chapter number 11.
Yes, ma'am.
How are you doing?
I'm doing pretty good. Staying home, staying healthy.
Good, good, good. I'm trying to do the same thing. I wish my
Yes,
house
ma'am.
could stay clean. My kids have gotten things everywhere, and I need to do a cleanup. But I think somebody coming to cut the grass today. But I got clothes everywhere. My kitchen, everything is just right now. So
Yes.
Uh, did you get the test and everything right? Everything was good
I
on your
did, yes. Ma'am.
okay.
Mhm.
All right.
I appreciate you saying
Oh, it is not a problem. I was just trying to make it easier for me and you guys, because I know y'all probably busy with other classes as well.
yes, ma'am.
Um, so I didn't want to stress you guys out too bad.
Mhm.
Uh, well, I'm gonna go on and get started. I don't know if anybody else is gonna join
Okay.
today. Um, hold on just a second.
Yes, ma'am.
Oh, all right, there you go. All right, we're going to get started. Let me make sure my screen. Katie, can you see my screen?
Yes, ma'am, I can.
Okay, all righty. Hi, Duncan. Hello. Yeah. Okay. All righty. Okay. So I'm gonna go ahead and get started. Why you turn the camera back on? Why you turn the camera back on? Me? Yeah. I don't know what I'm doing. I don't know how to use this. Am I going now? No, you're still on. Yeah, I can see. Okay, you good. You're good. Now. Okay. All right, so we're gonna get started with chapter number 11. Um, this is a short chapter, but I will upload because I have carbohydrates from actually organic chem, and so I'm gonna actually use some of those slides as well. I'm just gonna post those, but some of the stuff from organic will help you guys with carbohydrates as well because they go over this at the end. And I'm gonna upload a quiz for you guys to questions so to be ready for the last test. So we're gonna get into this. So carbohydrates, what's the deal with them and everything? So monosaccharides are the first one to get into. All right. So the first thing that we're going to get into is looking at monosaccharides. Now, what is the definition for carbohydrates? They are known as sugars or saccharides. They occur as monosaccharides, meaning that they are simple sugars. They can occur as small polymers. Those are disaccharides and trisaccharides. And then you have your larger polysaccharides. And those are sometimes called complex carbohydrates. The simplest sugars are 3 carbons. And we're going to move on to that. Monosaccharides are made from, look at this, aldehydes, and they form aldoses. So this is glyceride right here. So so this is one of them. So they are made from aldehydes. So Katie, one of the things that I wanted you to see is where in a way, I think it's good that you're taking both biochem and organic at the same time, because you get a chance to actually see where both of them kind of hinge together. Meaning that you get to see how aldehydes and ketones are made, but also get to see where that plays a part in when it comes to biochem. And so glyceride is just one of the main ones that you see there. And now, and then, so you got them also made from ketones. They form ketosis. So this is dihydroxyacetone. And so acetone is a ketone, and you got those. So what we're talking about now in organic is now what you're about to see is about to play an important part with biochem when it comes to carbohydrates. And I'm hoping that you don't get things crossed up or mixed up. You won't. I'll make sure you keep them separate. But this is just one of those things that you actually will see. Now, are there some common monosaccharides? You got ribose, glucose, and fructose. So those are some of your common ones. So just to talk about it a little bit, a sugar such as glyceride, it got a carbonyl group in it and is a part of an aldehyde, is known as an aldose. And a carbohydrate such as dihydroxyacetone, which the carbonyl group is a part of a ketone, is known as a ketose. In ketosis, the carbonyl groups occurs at the C2 or the second carbon. Now, monosaccharides can also be described according to the number of carbon atoms they contain. So 3 carbons are known as trioses. Tetrosis contain 4, pentosis. So just like in organic, when you name your carbon chain, you just look at how many number of carbons you have in there. That's the same thing. The aldopentose ribose is a component of ribonucleic acid, RNA. It's derivative to deoxyribose, occurs in DNA. So by far the most abundant monosaccharide is glucose, and glucose is an aldose. So that's the most common one. It is preferred, it's the metabolic fuel for most types of cells. So that's what it is. Now, one thing about glucose, it is a chiral compound because several of the carbon atoms bear 4 different substitutes. So they are all with glucose is chiral. So remember that. Most of your amino acids are achiral, glucose is chiral. So this is a chiral compound. And so with that, as a result, glucose has a number of stereoisomers, as do all monosaccharides. But the symmetric um, dihydroxyacetone is one exception. So several types of stereoisomer that applies to carbohydrates. So stereoisomers, when it comes to carbohydrates, that is all in there. So what you're seeing in organic plays a very important part of what you're seeing in biochem. And so that's what we're saying here. Most carbohydrates are chiral. And so that's what you're seeing here. And that's what I wanted to make sure that you guys understand. Most of them are chiral. Let's see. Now, with that, you got enantiomers there. Enantiomers, they can't be superimposed, but you got carbohydrates that differ in configuration at one of the carbons. They are called empimers. So empimers are simple. So here you have common monosaccharide glucose and galactose. Galactose is an epimer of glucose. So that's what you're seeing there. So if you look at them, everything pretty much look the same except for the carbon and the hydrogen, the carbon and the oxygen switch places. So that's the only thing of what it is. It's still hooked on to the carbon, it's just where they're at. They just, the configuration around one carbon atom really differs. So ketosis and aldosis both have empimeric forms, meaning that they have empimers of each other. So they have those. They're not interchangeable. So they just say, for example, if you have an enzyme whose active site accommodates glucose, it may not accommodate galactose at all. So just because of that one little change around that one atom, it will change a little bit. Now, one of the things that we're going to look at now is cyclization generates alpha and beta ionomers. So what is an ionomer? The alpha ionomer, this is a hydroxy group, lies on the opposite side of the ring from the, that is a methoxy group. So CH2OH group of the chiral carbon that determines the D or the L configuration. In the alpha ionomer of glucose, the hydroxyl group points down. In the beta ionomer, the hydroxyl group lies on the same side. And so ionomers in aqueous solution can interconvert between alpha and beta forms. And so with that, this is what you're seeing. Now here, what you're seeing is a Fisher projection of glucose. And in the Fisher projection, you have the horizontal bonds pointed out of the page and the vertical bonds pointed below the page. Glucose cyclizes to 6 member rings and it represented by a Hartworth projection, and in which the heaviest bonds point out from the plane of the page. The alpha and beta ionomers freely interconvert. So that's what it is. Cyclic sugars are represented as Hartworth projections. So that's what it is. So you've been exposed to Fisher projections, and then you also have Hartworth projections. And so you have both of those, and that's what you see. Now, with that, you can have monosaccharides. They can be, you can just put them different types of ways. You can derive them different types of ways. And one of the things that we're looking at here is you got glucose, and then when you add uh, methyl alcohol to it, and what'll happen is you can get basically alpha glucoside and beta glucoside. So you can do it different ways. So that's what you're looking at right here. And so they just did a reaction of glucose with methanol. Um, and it blocks the ability of the glucose to function as a reducing sugar. So the glycosidic uh, bonds formed between the anomeric carbon and the oxygen of methanol, it can have alpha or beta conformation of configuration. So that's what you have here. Now, what they say here is chemical reactivity, often to say using carbon containing solution. We have used this Benedict's reagent can distinguish a free monosaccharide called a reducing sugar from a monosaccharide when an anomeric carbon has already condensed with another molecule. So you can do that. And so that's what they're doing here. If you take glucose and you react it with methanol, the result is a non-reducing sugar. So that's what you produce there. Now, you can have phosphorylation and have phosphorylated monosaccharides. So here, what you see is you got phosphorylated monosaccharides. Um, and they can do different types of processes. So other metabolic processes replaces a hydroxyl group with amino group to produce an amino sugar. So it's a gloxoamine. Oxidation of a sugar's carbonyl group and hydroxyl group can yield uronic acids, and those are sugars that contain carboxylic acid groups. And reduction can yield something else, xylitol, a sweetener used in sugarless foods. So xylitol, you guys have heard of, um, um, and it's just different things. So what they're saying is basically, hey, you can take these sugars, you can add different things to it, and you can phosphorylate them, and they can become intermediates, and they can become pathways for breaking down glucose. So these are just two here, and this is the saccharide 3 phosphate and fructose 6 phosphate, and they can become pathways to actually break down those sugars. So you can have that. You can have this fructose 6 phosphate, and it can break down fructose. This glyceride 3 phosphate can break down glucose. So that's what they're doing here. You can phosphorylate them, and they can break down those sugars as well. That's what it is when you're taking medicine or like to get insulin to produce, because that's what insulin does, is basically help you um, process sugar, and that's what it is. Now, you got nucleic acid monosaccharides as well. You got ribose and 2-deoxyribose here as well. And with that, let me see here. What happens is one metabolically essential carbohydrate modifying reaction is the one that catalyzes ribonucleic reductase, which reduces the 2-OH group that is on there, that base of ribose, to convert it to a ribonucleotide to a diribose nucleotide for DNA synthesis. So this is what you can do here. The nucleic acid monosaccharides can reduce down and can do DNA synthesis. So that is what can happen. All right. So you got different types of monosaccharide derivatives. Glucosamine, we have heard of that one before. Um, Glucuronate and xylitol, you've heard of xylitol before. Um, so those are some common ones that you heard of. Now, let's talk about polysaccharides a little bit and what goes on with polysaccharides. Now, monosaccharides are those building blocks of polysaccharides. When you have those glycosidic bonds linked in successive residues. Unlike the amino acid and the nucleotides, the other polymer forming biological molecules, which are linked by one configuration, can be hooked together in a variety of ways. They can produce a lot of things. Each monosaccharide contains several free OH groups. And when they contain those several free OH groups, um, that permits different bonding arrangement, and that can allow for branching. So let's look at lactose. Lactose is this type of structure right here and everything. Carbohydrate chains or glycanes can be sequenced using mass spectrometry or mass spec, and the results can be ambiguous due to the inability to distinguish those isomers. So glycan and other three-dimensional structure are typically studied using NMR techniques. Um, so due to the challenges of this structure, glycomics, or the study of carbohydrates, is not fully developed as genomics or proteomics. So that's the thing. So glycomics, a lot of people are starting to get into that. So the most complex one is your oligosaccharides. Um, those are the ones, polysaccharides that are truly enormous, generally do not exhibit certain things and complexity of your other oligosaccharides. They tend to be one or more pair of monosaccharides that are linked over. Now, most common disaccharides. So let's take a look at glucose. Two monosaccharides to generate a disaccharide. In nature, disaccharides occurs as intermediates in digestion of polysaccharides, and that's the source of metabolic fuel. So you got lactose secreted into the milk of lactating mammals, consists of the lactose and glucose. And so that is what you have here. Um, that's what they have. And then some people, when they say that they are allergic to lactose or they are lactose intolerant, mostly what it is one part of this is not so it can be the whole thing of lactose. It may be a part of the lactose that is not good for them in the milk. So now they are starting to, like, in different things, remove that part, so that they'll know, so people won't be lactose intolerant. They can still get the things that they need out of milk. Now here is the sucrose one, or table sugar, is the most abundant disaccharide in nature. So sucrose is glucose and fructose together. So that's how it's linked. Sucrose is a major form which newly synthesized carbohydrates are transported from plant leaves, where most of your photosynthesis occurs, to other plant tissues to be used as fuel or stored as starch for later. Now, starch and glycogen are polymers of glucose residues. So let me just move on to that. They are storage fuel forms of glucose. So they are linked by glycosidic bonds designated as alpha 1 through 4. In other words, basically, the anomeric carbon 1 of the residue is linked to the alpha glycosidic bond to carbon-4 of the next residue. So plants manufacture a linear form of starch, that's amylose, which may consist of several thousand glucose residues. Amylopectin, an even larger molecule, can link 24 to 30 glucose residues to generate a branch polymer. And so when you're looking at carbohydrates, polysaccharides, disaccharides, monosaccharides, that is basically what you're looking at is how do we use these particular type of sugars for fuel. Now, animals store glucose in the form of glycogen. So this is the amylopectin structure here. And so it's larger, so 24 to 30 glucose residue structures. So cellulose. So when we get to cellulose, it's composed of glucose in beta 1 to 4 linkages. So earlier, when we talked about amylopectin structure, and that is, this is alpha 1 through 6, alpha 1 through 6, glycosidic linkages, every 24 to 30 glucose residues. Now for cellulose, this glucose in beta, and this is going 1 to 4 linkages there. So cellulose, like amylose, is a linear polymer containing thousands of glucose residues. However, the residues are linked by beta 1 to 4 rather than alpha 1 to 4. The simple difference in the bonding has profound structural consequences. What happens? Whenever starch molecules form compact granulars inside the cell, cellulose forms extended fibers that lend rigidity and strength to a plant cell walls. Individual cellulose polymer form bundles, extensive hydrogen bonding within and between adjacent chains. So the plant's cell walls include other polymers that together with cellulose yield a strong but resilient substance. So recovering carbohydrates from materials such as wood remains a challenge, especially for biofuels industry. So a lot of biofuels really been looking at this. Now, one thing about animals, they don't synthesize cellulose, and most of them cannot digest it in order to use this glucose as an energy source. So this is your cellulose structure. This is how it looks. Glucose residues are represented as hexagons. The carbon atoms are gray here, and then your oxygen are red. Not all the hydrogens, they are the small white circles, are shown. Hydrogen bonds are the dashed lines. They residues in the same adjacent chain, so a bond of cellulose polymers, that's what makes that makes them rigid. We're here, so this is biofilm from Pseudomonas and aeruginosa. So this is a pathogen bacteria growing on the surface of an agar plate. And that forms a biofilm with a complex three-dimensional shape. So bacteria also have polysaccharides. They form a biofilm. So prokaryotes, they do not synthesize cellulosic cells or store fuel as starch. So what they make is a biofilm. And so that biofilm basically forms and harbors that harbors all of that bacteria. And so that's what happens. So it looks like plaque that form on teeth, protect bacterial cells from washing away and protect from desiccation. Biofilms developed on medical apparatus such as catheters. And so they can be carriers of different stuff. Okay, I don't know why my kids are acting up upstairs, but hold on for just a second. Okay, I'm not going to worry about that. I'm going to move right along. So glycoproteins, and I think this is the last thing we're going to go over today. Chapter 11 is kind of short, but like I said, I'm going to upload some different things for you guys so you guys can have that. Like I said, we only got a few more chapters. I'm going to try to knock these chapters out real quick next week. Wow, yeah. So we can get this done because I want to go ahead and be um, done with this, um, with everything. So taking a look at the last part, which is glycoproteins. Now, there are so many different types of monosaccharides and so many ways to link them. The number structure for oligosaccharides with even just a few residues are enormous. So organisms, they take basically um, advantage of that, and that's what they do. So one of the things that we're going to look at is uh, proteins that are secreted from eukaryotic cells or remain on their surface are glycoproteins in which one or more oligosaccharides chains are covalently attached to polypeptide chain shortly after it is synthesized. So what you're seeing here is oligosaccharides are attached to glycoproteins are usually linked either to a side chain, so end-linked oligosaccharide, or to a serine or threonine side chain, and that'll be O-linked. So end-linked or O-linked. So glycoproteins, they're going to be O-linked or end-linked. And so what you see here is end-linked right here. And this is your O-link right here. And so serine or 309. So that's what you see here. So in wait, let me go back with that. Glycosylation begins when a protein is synthesized by a ribosome associated with the rough endoplasmic reticulum, which is the ER. So that's what you're seeing right here. So what is going on is glycosidasis and glycotransferase, and that happened in the Golgi apparatus process. That is the 14 residue oligosaccharide that is attached to the newly synthesized protein in the ER. The 5 residue core oligosaccharide with 3 monos and 2 N-acetylglucosamine residues. Those are in the center. Is common to all end-linked oligosaccharides. Only one of the many possible mature oligosaccharides is shown. So how do we go through all of that? So that's what you see here. So you see all the different ones. You see the nucleosynthesized one right there is attached to the protein. That's trimmed by the glycosidasis. Then partially processed glycoprotein. It has the core and then you got additional monosaccharide residues are added, and then you get your glycoprotein, and then your oligosaccharide will be matured then, because it occurs during that synthesis. Okay, sorry that the kiddos are crying. So you got the structure of an end-linked oligosaccharide. This is 11 monosaccharide residues linked by glycosidic bonds. Experience considered conformation flexibility. So the structure here is just one of many that is possible. So if you see the arrow down there at the bottom, that just shows you where the end atom of the ASN side chain, where the glycan is attached. So it can attach there. Now, what can oligosaccharides do? Because they are highly hydrophobic and conformationally flexible, they can accept or occupy a large effective volume above the protein surface. So they can be used as biomarkers, biological markers. Now, you can have proteoglycans, and they have chondroitin sulfate on there, and they can have repeating units of that. So these are glycoproteins in which protein chains serve mainly as an attachment for an enormous O-link polysaccharide. So that is called glycosaminoglycans. And so most of them contain a repeating unit of a disaccharide of a amino sugar. And those are N-acylated. And so that's what you see in the blue right there. And so this sulfate contains 100 of these disaccharide units, and the degree of sulfation may vary along the length. Now, when it comes to bacterial walls, they are made of peptidoglycan, peptidoglycan. That is, it surrounds the plasma membrane of the cell and determines the overall shape. The carbohydrate component in many species is repeating beta 1 through 4 length disaccharide, and usually the length is probably 20 to 40 disaccharides. And so what you see in here is E. coli. In this model, looking down on the cell surface, is a single layer of the peptidoglycan shown with the carbohydrate strands in blue. So this is what you're seeing here. And so peptides are 5 or more amino acids. They covalently cross-link the saccharide chains in 3 dimensions to form a structure really thick. Antibiotics of the penicillin family blocks formation of peptide cross-link, thereby killing bacteria without harming their eukaryotic yolk. Archaic bacterial walls contain proteins and sometimes polysaccharides, but not peptidoglycans. So that's what you see there. Basically, this is it. What is it that I want to say for this chapter? Carbohydrates have a general formula that is CH2O. N exists as monosaccharides and polysaccharides of different sizes. The monosaccharide may be aldeoses or ketoses and exist as enantiomers. Those are mirror images and as epimers, which are different in configuration at the individual carbon atoms. Cyclization of monosaccharides produce alpha and beta anonomers. Formation of a glycosidic bond prevents the interconversion of alpha and beta forms. Monosaccharide derivatives include phosphorylated sugars, sugars with amino, carboxylic, and extra hydroxyl groups, and deoxy sugars. Okay, when it comes to polysaccharides, lactose consists of the lactose-linked beta 1 to 4 glucose. Sucrose is glucose-linked alpha 1 to 2 to fructose. Starch is a linear polymer of alpha 1 to 4 linked glucose residues. Glycogen also contains alpha 1 to 6 branch points. Cellulose consists of beta 1 to 4 linked glucose residues. In trypsin, the residues are N-acetylglucosamine. A bacterial biofilm is a community of cells embedded in extracellular polysaccharide matrix. And the last thing that I want to say is oligosaccharides are attached to proteins. They are N-linked or O-linked. N-linked undergo processing by glucosidases and glucosyltransferases. The carbohydrate chains of the glucoproteins as protection as recognition markers. Proteoglycans consist of long chains that can function as shock absorbers, lubricants, and protective coatings. And your peptidoglycan in bacterial walls is constructed of cross-linked oligosaccharides and peptides. So that is all for this chapter. This weekend, I'm going to try my best to, because I got some other things that are due that are outside of school, to work on getting your, what is that thing called? Your rubrics up for your different papers and stuff. So I'm going to try to make sure I get that done this weekend and everything. Katie, if there's anything else, if you guys need, just let me know. Send me an email, but I'm going to try to get them up this weekend so you guys can have it. Next week, we're going to be going over chapter 12 and 13. I'm going to try to wrap up everything next week. That is my goal. Try to get 13. So 12, 13, and 14. I'm going to try to get mostly everything wrapped up next week. I do have some quizzes that I am going to post for you guys. So I'm going to post those quizzes to help you guys for the last test. So I'm going to try to get everything put on early so you guys can have those and everything. That's all I have for today. That's it. Like I said, send me an email. Stay safe. I will talk with you guys later on. Have a good one.